Hello everyone, this is Venkatesh, first MSc Microbiology from Sacred Heart College, Tirupattur. In this video lecture, I am just making you to understand about virus structure and classification. Let's we have a closer look on the topic. General characteristics about virus. Viruses are acellular in nature. Why? Because it lacks cell wall and cell membrane. In addition, it doesn't contain cells and which is not having the property to divide like other cells. So that's why it is referred as acellular in nature. Viruses that are oftenly measured in nanometer or and the sizes ranges from 20 nanometer to 1500 nanometer. The viruses that are oftenly called as living chemicals, which means in outside of the host they do not grow or reproduce by themselves. However, when a virus enters into the living cell, it obtains energy from the host cell and starts reproducing. So that's why it is considering as living chemicals. Then the viruses are obligate intracellular parasites. Here the obligate representing strictly. So the virus must need a host system for the replication. It only possesses the replicates inside the living cells alone. The viruses that can be observed by means of electron microscope, viruses are filterable agents. The virus that contains nucleic acid, either it can be DNA or RNA, but never be the both. Viruses that lack cellular organelles such as mitochondria and ribosomes. Viruses that causes disease in human, plants and animals. In humans, AIDS, SARS, polio, common cold. These are all the some viral infections that are caused by virus in humans. Whereas in plants, the tobacco mosaic virus that causes the disease in tobacco mosaic plant. In papaya, it causes papaya ring spot disease. Father of virology is Martin William Beijerick is considering as the father of virology. Why? Because at the 19th century what happens, his tobacco plant was affected by disease and he thought that it might be a bacteria or toxins. So he filtered sap of the diseased plant against the bacteria. Then he injected the sap of the diseased plant into healthy plant. The plant gets sick. So he concluded that a smaller pathogen than the bacterium could pass through the filter. So he initially called it as Contagium vivum fluidum, but later it turned as virus. So that's why he is considered for as father of virology. The locomotion, regarding the locomotion, the viruses are non-motile, so which is not having the property to move one place to another place. The study of virus is called virology. The structure of virus. The virus that are composed of nucleic acid, as I told before, it contains any one of the nucleic acid. Either it can be DNA or RNA that are surrounded by a special protein called capsid. Some viruses have enzyme in the genetic material that is reverse transcriptase, which is commonly found in the reovirus. Based on their capsid arrangement, the viruses that are classified into four major types. The helical form. If the arrangement of the capsid protein in helical form, then it is having helical structure. The well-known example is tobacco mosaic virus. Likewise, if the arrangement of the capsid protein is polyhedral or icosahedral form, then it is having polyhedral or icosahedral structure. Adenovirus is well-known example for icosahedral structure. Likewise, influenza virus is having spherical form and bacteriophage is having complex viral structure. Now we shall discuss the basic structure of a virus. Here I have listed out some viral particles that are commonly found on the structure of virus. The first is capsid. The capsid is a protein shell or a coat that encloses the nucleic acid, which means it protects the genetic material of the virus. The second, capsomere. The capsomere that representing polypeptide that are made up of identical protein subunits. Nucleocapsid. The combination of capsid with nucleic acid that are surrounding the genetic material of the viral genome and commonly found in the complex virus such as bacteriophage. The fourth is envelope. If the membrane of the viral structure that is made up of lipid, it is referred as the envelope. The virus which are lacking the envelope are called naked virus. Spike. Spike is a kind of protein that are made up of glycoprotein which is commonly found on the surface of the envelope. So which is essential for the attachment of the virus into the host cell. Here I would like to mention one thing. A definite vaccine is not made so far to the coronavirus due to the spike protein of the virus is keep on changing. The spike protein here, the spike protein that determines sometimes the virulence or the mutation of the some viruses. The well-known example for that is coronavirus. The last viral particle is viral genome. As I told before, the virus that contains either of the nucleic acid, whether it can be DNA or RNA, but never contains both. So some viruses I have told you that possess the reverse transcriptase enzyme in the nucleic acid. So that are possessed by all single-stranded RNA virus which are having the negative sense strand. Next, the classification of virus. There are four types of viral classifications existed. The first is Holmes classification. So he classified the virus on the basis of Carolus-Linnaeus binomial nomenclature system. The second classification system is LHT system. So LHT that representing three scientists, they are namely Louf, Horn and Tournier. In the year 1961, they classified the virus on the basis of nucleic acid, symmetry, envelope and capsid arrangement. Third classification system, ICTV, which stands for International Committee on Taxonomy of Virus. So they classify the virus on the basis of taxonomy hierarchy like kingdom, phylum, class, order, genus, species such as. The fourth classification and standard classification is Baltimore classification on the basis of nucleic acid. So in the year 1971, virologist namely Baltimore, he classified the virus into 7 major groups on 4 major bases. The first major characteristic is nucleic acid, based on whether the virus is having DNA or RNA. The second difference is strand, whether it possesses single strand or double strand. Third difference is sense of the strand, whether it possesses positive or negative strand. The fourth and last is mode of replication. So based on these four major characteristics, he classified the viruses into 7 major groups. So this is the Baltimore classification. Here I have listed out the 7 major group of the virus that are classified on the basis of nucleic acid. The first group is the virus which is having the double-stranded DNA. The example includes adenovirus, herpes virus, pox virus and papilloma virus. The second group, the virus that having single-stranded DNA. Well-known example is parvovirus. Third group, the virus is having double-stranded RNA. Reovirus is the example. Fourth group of viruses which is having single-stranded RNA with positive sense. Examples include poliovirus, togavirus, hepatitis A and B, C virus. Fifth group viruses which is having single-stranded RNA with negative sense. Examples are influenza virus, rhabdovirus and orthomycoviruses family. Sixth group, the viruses which is having single-stranded RNA with reverse transcriptase enzyme. The well-known example of the single-stranded RNA with a reverse transcriptase is HIV virus. The last group of viruses that possess double-stranded DNA with reverse transcriptase enzyme. Example is hepatitis B virus. With this, the virus structure and classification is explained. So thanks for your pleasant listening and catch you on next interesting topic. Thank you.